information. So how many chromosomes do we have inside the nucleus?
23 pairs?
Yeah, very good. We have 23 pairs, so a total of 46 chromosomes, which is being numbered from chromosome 1 to chromosome 23. From chromosome 1 to chromosome 22, these are classified as autosomal, okay? While the last pair, the 23rd chromosome, is the sex chromosome. So if you have a pair of two X chromosome, then the sex will be female, while the 23rd, if it is X and Y, then the sex is male. So this will distinguish the sex, okay? Whether you have the Y chromosome or both the X chromosome. If this particular condensed chromosome actually, okay? So if this is being extended, as you can see, it is a condensed form. But when you extend it, it is a long chain of DNA molecule, okay? And this is what we're going to discuss today, the composition of the DNA molecule. So looking on the upper box here, the top portion of your slides, you have different color-coded bars, okay? The green, yellow, red, blue. And these four colors actually represent the four repeating nucleotides in a DNA molecule. And probably by now, you are aware of the four letters which will spell the DNA molecule. What are these four letters? A, G, C, and T. So these bars, the four coded bars, actually represent the four letters. Okay, and then as you can see, this DNA molecule coils around a protein. Okay, and these are the histone proteins. Okay, histone proteins primarily function in packaging of the DNA molecule. Okay, so as you can see, the DNA molecule coils around the histone. Okay, and then this will further coil. Okay, so there will be supercoiling until you form the condensed chromosome. Okay, so the lower part of your slide represent the condensed chromosome, but when it is unpacked and extended, it is a long DNA molecule. Okay. So again, the function of nucleic acid, it is considered as informational molecule, primarily genetic information. And we have two kinds of DNA molecule, or rather nucleic acid. You have the DNA and the RNA, okay? So the DNA is the deoxyribonucleic acid, while RNA is ribonucleic acid. Between the two, DNA is considered as the blueprint of living organism, primarily because it has the genetic information which is needed for the synthesis of proteins, while RNA usually acts as machinery for the expression of the coded information in DNA to proteins. And an example of RNA that carries genetic information is known as mRNA. M stands for messenger RNA. So mRNA is considered as the carrier of genetic information encoded in the DNA. But there are other organisms whose genome is an RNA molecule. Example will be viruses, okay? Like the human immunodeficiency virus, okay? The causative agent of AIDS. The genome is an RNA. So if I ask you in the exam, which among the following is considered as carrier of genetic, or rather storage of genetic information, then you can answer both DNA and RNA. But eukaryotic organism like human beings, DNA is the blueprint or it contains genetic information, while mRNA is the carrier of genetic information. Okay. And then another important concept in relation to nucleic acid chemistry is the central dogma of life. Okay. So what is this central dogma of life? Sorry. The central dogma of life is considered as the flow of genetic material. This is how the information is transferred from DNA, okay, to mRNA to protein, okay? And there are three important processes under the central dogma of life, namely, one, first, replication, second, transcription and translation. So replication, okay, is actually the synthesis of identical DNA molecule. That's the reason why it is called replication from the word replica. You will be producing identical DNA molecule, okay? While transcription is the synthesis of RNA. And the template for you to synthesize RNA is the DNA molecule. So this is DNA to RNA. Not only mRNA, because we have also tRNA and ribosomal RNA. While the last process under the central dogma of life is translation. It is the synthesis of protein from the mRNA template. The word translation is you convert one language to another language. Basically because RNA is composed of nucleotides while protein is composed of amino acids. So you have two different languages here. Okay? Then what about the covalent structure of nucleic acid? Okay? So genetic information is encoded in DNA and carried by mRNA as sequence or a chain of nucleotides. Okay. So what is a nucleotide? A nucleotide actually consists of base, sugar, and a phosphate group. Okay. A base is actually a nitrogenous base, which may be a purine or a pyrimidine. Purines are adenine and guanine, while pyrimidine is cytosine and thymine. For the sugar, it contains a 5-carbon sugar, so you call it pentose. And it could either be ribose or deoxyribose. Okay. The ribose is the sugar found in RNA, while deoxyribose is the sugar found in DNA. And then this base and sugar is esterified to a phosphate group. Okay. So what's the difference between a nucleoside and a nucleotide? A nucleoside is just a combination of a base and sugar, while a nucleotide is a combination of base, sugar, and a phosphate. Or it is simply a nucleoside which is phosphorylated. Okay? So that's a major difference between a nucleoside and a nucleotide. Again, nucleoside is composed of base and sugar, while nucleotide is composed of base, sugar, and a phosphate group. Okay? So these are the nitrogenous bases divided into purines and pyrimidines. So again, purines, adenine represented with the single letter A, and guanine represented with the single letter G. While we have 3 pyrimidines, cytosine, C, uracil, U, thymine, T. Cytosine and thymine can be found in your DNA, while uracil and cytosine is in your RNA. Then for the sugar, again, deoxyribose for DNA and ribose for RNA. In a nucleotide, the sugar hold the base on one side and the phosphate on the other side. So the sugar is at the middle. Okay. So the sugar hold the components of the nucleotide together. So again, this is your base attached to a sugar. So a combination of sugar and base will form now a nucleoside. And these two can be linked via, you know this already in carbohydrate chemistry, this is what you call and glycosidic bond. Okay. So again, the sugar has this numbering. This is carbon 1. 2, 3, 4, and 5. So the first carbon is always linked to a base, while the fifth carbon is linked to your phosphate group, to the phosphate group. Okay? And the linkage is known as a phosphoester linkage. This is the second carbon. The importance of the second carbon is when you have the OH group here, this is a ribose sugar. So this will be the nucleotides for RNA molecule. But if the second carbon has only hydrogen, so this becomes a deoxyribose. So without the oxygen, this will be the nucleotide repeating units for DNA molecule. And then this will be the third carbon with the OH group. Okay? So again, these are the following nucleotides in DNA. First, adenine 2-deoxyribose phosphate, guanine 2-deoxyribose phosphate, cytosine 2-deoxyribose phosphate, and only found in DNA, thymine 2-deoxyribose phosphate. While the nucleotides in RNA, adenine ribose phosphate, guanine ribose phosphate, cytosine ribose phosphate, and for RNA, we have uracil ribose phosphate. Okay. So of course, the difference between DNA and RNA, again, the sugar deoxyribose for DNA and thymine. While for RNA, you have the ribose and uracil. Okay. So this is the chemical structure of a nitrogenous base. For purines, it consists of 2 rings, okay? 2 rings. And these are the numbering, okay? While pyrimidine, it has only 1 ring, okay? So purine is bigger than pyrimidine. But when you cut this part, okay, when you cut this part, it actually resembles pyrimidine, okay? Like this. So what are the pyrimidines again? Cytosine, uracil, and thymine. So it's easy to remember why. Because pyrimidines are cat purines. Cat stands for cytosine, uracil, thymine. So you just remember the other 2, which are classified as purines. Okay. Wait. Sorry for the interruption. So again, just to differentiate purine from pyrimidine, purines will be bigger with 2 rings, while pyrimidine is just 1 ring. You cut this part, that becomes pyrimidine. Okay? So again, what are the pyrimidines? Cytosine, uracil, and thymine. And what are the 2 purines? Adenine and guanine. Then you can differentiate one base from another base by knowing the groups attached on that particular ring, okay? So it can be oxygen, amino groups, or methyl group. So for purines, okay, adenine has a chemical name of 6-aminopurine, meaning there's amino group attached to the 6th carbon. While for guanine, the chemical name is 2-amino-6-oxypurine, wherein on the 2nd carbon, you have an amino group, while on the 6th carbon, there is oxygen. For the pyrimidines, first you remember uracil. The chemical name of uracil is 2,4-dioxypyrimidine, meaning you have 2 oxygen attached to pyrimidine at carbon 2 and 4. Then you substitute amino group on the 4th carbon, making it 2-oxy-4-aminopyrimidine. This becomes cytosine. For thymine, you add another methyl group on the 5th carbon to uracil, 2,4-dioxy-5-methylpyrimidine, then this time it is thymine. So you can differentiate one base from another base by knowing the groups attached on the rings mentioned for the purine and pyrimidine. So again, this is your purine with 2 rings. Adenine, what is the chemical name of adenine? This is the 6th carbon. 6-aminopurine. How about guanine? What is the chemical name of guanine? This is the 2nd and the 6th carbon. 2 amino
Amino 6-oxypurine.
purine. Okay? So, how about the pyrimidines? First, uracil. Start with uracil. This is the second and the fourth carbon. So what is the chemical name of uracil?
2,4-deoxypyrimidine.
2,4-dioxypyrimidine. Okay? How about cytosine? 2-oxy 4-aminopyrimidines. How about thymine?
2,4-deoxy-5-methylpyrimidine.
2,4-dioxy 5-methylpyrimidine. Okay? So again, a nucleoside is composed of base and sugar. And this is linked with N-glycosidic bonds. Specifically, for purines, the linkage is N9C1. For pyrimidine, the link is N1C1. Okay? So always, the sugar at carbon 1 is linked to your base. If it is purine, it is linked to N9. Okay? Well, for pyrimidines, it is linked to N1. Okay? So you have adenine linked to a sugar, the name becomes adenosine. Guanine plus the sugar ribose, it becomes guanosine. So usually, nucleoside ends with SINE. But for pyrimidines, take note, cytosine, even though it ends with SINE, this is still a base. So unlike here, adenosine and guanosine. The nucleoside counterpart of cytosine is cytidine. For thymine, thymidine. Nucleoside, it ends with DINE. So take note of this, okay? Because cytosine is still a base. The nucleoside counterpart of cytosine is cytidine, okay? So again, can you identify this part on the left side of your slide? Oh, what is this? First, you identify the base. What is the base? Anyone? Can you recognize this? Is this a pyrimidine or a purine? Pyrimidine. What type of pyrimidine? Only one oxygen. What is that?
Cytosine.
Cytosine. Then you add the sugar. From cytosine, it becomes what?
Cytidine.
Cytidine. Very good. How about this one on the right side? What is this base? With an amino and oxygen.
Guanine.
Then you add the sugar. What is the nucleoside? What is this nucleoside?
Guanosine.
Are you sure it's guanosine? Take note of this part. This is the second carbon. Is it guanosine? Any other?
Guanine?
What?
Guanine?
Guanine? You're right, it's guanosine, but is it guanosine? Is it just plain guanosine? Deoxyguanosine, okay? So it should be deoxyguanosine. And then, from nucleoside, it can be converted to a nucleotide by adding phosphate groups. So you can add 1 phosphate, 2 phosphate, or 3 phosphate groups, okay? And the abbreviation for the nucleotides can be AMP, that stands for adenosine monophosphate, 2-phosphate adenosine diphosphate, and 3-adenosine triphosphate, and so on, okay? So this is the acceptable shortcut for these nucleotides. So this is the structure of the most famous molecule in biochem, which is the energy currency of the cell. You were able to discuss this under energy metabolism. So if I ask you now, can you write the structure of ATP? Can you do that? ATP has the base, adenine, attached to sugar ribose, that becomes adenosine, plus 1 phosphate, this is now adenosine 5' monophosphate, 2 phosphates, adenosine 5' diphosphate, and 3 phosphate groups, adenosine triphosphate. So the entire structure now is the ATP molecule, okay? So nucleotides can be linked with one another. By what bond? It is linked by a phosphodiester bonds, okay? And the link is between the 3' hydroxyl on the sugar of one nucleotide and the 5' phosphate on the sugar of another nucleotide, okay? So you can have tri, 4, okay? So that's how you link the different nucleotides. And take note, these compounds have polarity of direction, okay? And later in the structure, I'll emphasize this. So the polarity of direction is 5' to 3'. Because it has a phosphate group, the nucleotides are acidic, okay? So it has a DNA, the DNA has a net negative charge, okay? So this is what I have explained. One nucleotide plus another nucleotide, the linkage is between the 3', okay, 3', this one, 3' hydroxyl group, and the 5' phosphate, okay? So 3' hydroxyl group of one nucleotide added to the 5' phosphate of the next nucleotide. And you form what linkage? Phosphodiester linkage. In contrast, this is a phosphoester linkage, okay? This one is a phosphodiester linkage, okay? And the polarity of direction runs from 5' to 3', meaning you can now check the sequence of nucleotide starting from the 5' end to the 3' end. If you reverse the sequence, then it will be abnormal, okay? So you need to take note of the polarity from 5' end going down here up to the 3' end. So if I ask you, what is the sequence of that DNA molecule? 5', thymine, adenine, cytosine, guanine, TACG, okay? So this will be your base, the sugar, and the phosphate group. The nucleotides connected with one another via a phosphodiester linkage between a 3' hydroxyl and a 5' phosphate. So the incoming nucleotide is attached to the 3' hydroxyl group, okay, via a phosphodiester linkage. And then, knowing the structure of the 4 nucleotides, you can generate other structure from these nucleotides. Example, ATP can be converted to cyclic AMP. Again, this molecule, cyclic AMP, is a very important molecule in biochem. The function of this is a second messenger. So how did we generate cyclic AMP? So again, this is your adenine attached to ribose, and you have here one phosphate, and this phosphate is linked again to the 3' hydroxyl group. So meaning from ATP, when it is converted to AMP, the 2 phosphates will be removed, and the remaining phosphate groups will be linked to the 3' hydroxyl group. So this is the structure of cyclic AMP, okay? To complete 3', 5' cyclic AMP, a very famous second messenger. In addition, you have also cyclic GMP, another second messenger. Same with AMP, it is derived with GTP. So from GTP, you remove the 2 phosphates, and then phosphate linked with the 3' hydroxyl group, forming 3', 5' cyclic GMP. And then this one is S-adenosyl methionine. What is the function of S-adenosyl methionine, which is abbreviated as SAM? First, it is composed of adenosine, okay? So you have adenine, and sugar ribose, linked to amino acid methionine. Function of SAM, it is considered as a methyl donor. So it usually participates in methylation reaction, okay? Then other derivatives of nucleotides will be the following, which you already encountered under energy metabolism. So you have NAD, nicotinamide adenine dinucleotide. NADP, nicotinamide adenine dinucleotide, okay, phosphate. FAD, okay, it's riboflavin or flavin adenine nucleotide and coenzyme A, okay? So these are your derivatives of nucleotides. So these coenzymes are actually derived from vitamins, okay? Nicotinamide, that is your vitamin B3. Riboflavin is vitamin B2, and pantothenic acid is vitamin B5. This is the reason why you need to take your vitamins either from supplement or from fruits, because these vitamins are involved in energy generation, which you have already discussed under energy metabolism. Again, the second messenger cyclic AMP, SAM, okay, as adenosyl methionine. And then there are selected synthetic pyrimidine and purine analogs. So these are look-alike of purine and pyrimidines, which are being used as drugs. For example, in the treatment of gout, okay, when you have excessive uric acid, the name of the drug is allopurinol. And then example of drugs used in the treatment of cancer, 5-FU, okay, mercaptopurine, thioguanine. So if these chemotherapeutic drugs will be administered to a cancer patient, they will be a look-alike of your purine and pyrimidine. So when added in a growing DNA molecule of a cancer cell, then the DNA becomes abnormal. And that is usually the goal in chemotherapy, to kill the cancer cell by adding a pulse nucleotides. And these are the pulse nucleotides added during DNA synthesis in a cancer cell. This is another drug, antiviral drug, arabinosyl cytosine or cytarabine and azathioprine. And they are look-alike of nucleotides. Okay. So again, to differentiate RNA from DNA, the sugar is deoxyribose in DNA and ribose we have for RNA. Okay. And the bases will be guanine, cytosine, adenine, and uracil. Okay. So you can easily remember this, the difference between DNA and RNA. These are the different ways of presenting the structure of a polynucleotide. So take note, you have the polarity of direction from 5' to 3'. So it can be written as single letters. So just to note the sequence, TACG, G, G, and so on. Or you can include in between the phosphate group because of the attachment, the phosphodiester bond. Or you can even draw the structure, okay? So this is what you call the sugar phosphate backbone. And then these are the bases. So the bases now will spell the genetic information. Okay. So as proposed by Watson and Crick, okay, the DNA as the genetic material is actually composed of double-stranded DNA, okay? And it was actually proposed in 1953. So you have here two anti-parallel ribonucleotide chains, one around each other with bases inside the helix, sugar, and the phosphate, okay? So the bases are inside while the sugar phosphate are on the outside. It is also described as stacking of the hydrophobic rings. Hydrophobic rings refers to the bases, okay? And the bases, you have two strands, okay? The two strands are connected by a hydrogen bonding of the bases. And you have the base pair, thymine and adenine, while cytosine is paired with guanine. For thymine and adenine, this is connected with two hydrogen bonds, while for cytosine and guanine, you have three hydrogen bonds. Another description proposed by Watson and Crick, the two strands of polynucleotide run anti-parallel. What do you mean by anti-parallel? One strand runs from 5' to 3' end, while the other strand is from 3' to 5' end. So they run in opposite direction. Okay, so this is the sugar phosphate backbone on the outside. Okay, sugar phosphate backbone on the outside. And on the inside of a double-stranded DNA, you have the hydrophobic rings, paired adenine thymine and cytosine guanine. They are attached by hydrogen bonding. So if you hydrolyze the hydrogen bonds, the double-stranded DNA becomes single-stranded. Okay. So you have more hydrogen bonds between cytosine and guanine, while for thymine, you have only 2 hydrogen bonds. And this will explain why you have a stable pair for cytosine and guanine compared to thymine and adenine. Okay. Then according to Chargaff's rule, the content of A is equivalent to content of T, while the content of G is equivalent to content of C. So in the laboratory, if you're going to analyze the DNA molecule with its composition of nucleotide, you can just simply analyze one base and you can predict the other 3 bases. Example, in a particular DNA sample, if the content of A is 20%, what is the content of T? 20%. How about G?
80%.
80? Sabi ko, A is 20. T is 20. Anong remaining na lang? 60 divided by 2. So 30.
30.
Right? So you need to have a total of 100. So 30% for G, 30% for C, 20% for T, and 20% for A. And actually, Chargaff's rule is based on an experiment, okay? In order to establish that the actual pair is A and T, G and C. So you only examine one base and you can predict the other 3 bases, okay? Furthermore, the helix is described as 2 nanometer in diameter using electron microscopy, okay? And the bases on top of one another is separated by 3.4 angstrom. Okay, so ito yon. Rotation is 36 degree. So for every helical turn, you have 10 base pairs. So a total of 34 angstrom. Okay. But actually in a solution, it could be 10 or 11. Okay. So again, the function of DNA molecule is the storage of genetic information. How do you store the genetic information? A sequence of bases along the chain. So it will be the sequence of the 4 nucleotides. Okay. So example here, on one strand, you have T, G, Z, T, G, T, G, and G. You can predict the sequence on the other strand. T, A, G, C, C, G, T, A, G, C, T, A, G, C, G, C. Could be 5 prime, 3 prime, 3 prime, 5 prime. Okay? So this is the description for a double-stranded DNA. And for the secondary structure of the DNA, okay, which is the double helix, there is a minor group and a major group, okay? This is also important to emphasize because this group is where the proteins needed for gene expression, okay, this is the site for the attachment of proteins, which we considered as transcription factors, okay? So if you have the secondary structure of DNA, which is the double helix, what is the primary structure of DNA? What do you think is the primary structure of DNA molecule? The nucleotide sequence, okay? So the nucleotide sequence will be the primary structure while the secondary structure is the double helix, okay? So this is what you call the replication port. So from double-stranded DNA, it can be separated into single-stranded DNA. And the single-stranded DNA can act as a template strand for the synthesis of new DNA molecule. So this is what you see during replication. So during replication, normally the double-stranded DNA will separate as single-stranded. So it can act as a template for the synthesis of new DNA, okay? So this is DNA replication, which we considered it as semi-conservative. Why semi-conservative? Because the newly synthesized DNA consists of a new strand and an old strand. So this is how we describe DNA replication. It is semi-conservative, okay? So from this parental strand, you will be producing several molecules of DNA, an identical DNA molecule, okay? So this is the template and the growing strand. So during replication, the template strand runs from 3' to 5', while the growing strand, anti-parallel, runs from 5' to 3'. Okay? So the bases will be added here based on the template strand. So for example, if on the template strand you have the C, then what is the complement of C? G. If it is T, the complement is A. So you can predict the sequence of the growing strand by knowing the sequence of the template strand. You just follow the Watson-Crick base pairing rule, okay? And during DNA synthesis, okay, actually the substrate will be these DNTPs or DXTPs, okay? These are your deoxynucleoside triphosphate. So during replication, the two phosphate groups will be cleaved off. Only one phosphate will be left behind, which will interact now with the 3' hydroxyl group here, okay? So remember, you form a phosphodiester bond between a 3' hydroxyl and a 5' phosphate, okay? So the direction of replication actually runs from 3', okay? So it is from 5' to 3' rather, okay? So the 3' hydroxyl is always vacant to accept the incoming nucleotide, okay? So all types of nucleotides here are monophosphates because the two phosphate groups will be cleaved up during replication. So this is the typical secondary structure. It's a double helix and the twist is to the right. Okay? And I think in your exam, left-handed is wrong. It should be right-handed helical structure of DNA. So the twist is to the right. How about the tertiary structure? So if the primary structure is the nucleotide sequence, the secondary structure is the right-handed double helix, the tertiary structure of DNA is actually DNA supercoiling, okay? So in a prokaryotic DNA, you can have positive and negative supercoils. Supercoiling in eukaryotic DNA is very important to form the chromatin, okay? So basically, this is for packaging of DNA inside a nucleus, okay? And remember, DNA is negatively charged, so there will be electrostatic attraction with a positively charged protein, and that is your basic protein histone, okay? So again, this is the DNA molecule coiling around the blue circle. This is your histone. So histone are basic proteins, positively charged, interacting with a negatively charged DNA molecule, okay? Another concept in DNA is denaturation of DNA, okay? So denaturation of DNA here, okay, it is actually a normal process. Take note, the double helix is disrupted during DNA replication, transcription, repair, and recombination. So unlike in protein, when you denature the protein, there is loss of function. But for DNA, it's a physiologic reaction. Denaturation of DNA is a physiologic reaction, which is important during these processes, replication, transcription, and so on. So therefore, the forces that hold the two strands together are adequate for providing stability, but weak enough to allow easy strand separation. And the method of choice for DNA denaturation in the lab is actually alkali. Okay? Kasi alkali, such as sodium hydroxide, the OH group can interact with the hydrogen in the double-stranded DNA. And that's how you disrupt the hydrogen bonds, making the double-stranded DNA single-stranded. aside from alkaline, another factor that can cause separation of a double-stranded DNA is increasing temperature, okay? So you can disrupt the double-stranded DNA. And the temperature is actually known as the melting temperature, or TM. So what is TM? This is the temperature at which 50% of the double helix is unwound. So melting causes unstacking of the base pair, so separation of the base pairs, which result in increased absorbance or hyperchromic effect. Okay. So later I will explain that hyperchromic effect. And the TM is strongly influenced by the base composition of the DNA. So which do you think will require high TM values? DNA with more of 80 pairs or DNA with more of the CG pairs? What do you think? Which will require higher temperature to separate the double-stranded, to make it
ICG po.
GC pairs. Because GC has 3 hydrogen bonds, while 80 has only 2 hydrogen bonds. Okay? So higher TM for GC pairs than 80 pairs. For mammalian DNA, which is about 40% GC pairs, it has a TM value of about 87 degrees centigrade. In contrast to denaturation, another process is renaturation. You can call it also as annealing or reassociation. So again, if the temperature of the melted dissociated duplex DNA is rapidly reduced, for example, from 94 down to 70, the original double helical structure, okay, it does not reform, okay, but if you lower it by 20 by 25 degree below the TM, so example, 87 is the TM value, you lower it by 20, 67 na lang, then the original double helical structure reform. Okay, so first you need to know the TM and then lower it by 20 to 25 degree centigrade in order to have the single-stranded DNA renature. So this is the illustration. This is the native double helix. You reach the TM value of 87 degree for mammalian DNA. Half of the strand is unwound, single-stranded. Half of the strand is intact. More heat from 87, you raise it to 94 degrees centigrade, and completely you have denaturation of DNA, separated strands, single-stranded DNA. You cool it down, okay, to 67. So from TM of 87 minus 20, so 67, and expect the single-stranded DNA to renature, or there will be annealing back, okay? So a while ago, I mentioned about the hyperchromic effect. When the DNA molecule, okay, is completely separated, the bases will be exposed. So to demonstrate hyperchromicity effect, you need to use the spectrophotometer. Example, you have a tube containing a native intact double helix DNA. Okay, then you measure it in the spectrophotometer. Okay, the one that absorbs light are actually the bases. So this will absorb light using the spectrophotometer. And in a native DNA, the bases are covered. So expect that there will be less absorbance. But after heating, and the DNA is completely separated, the bases will be exposed. So these bases will be exposed. They have more chance to absorb light using the spectrophotometer. So expect that the absorbance reading can increase. And this is what you call as the hyperchromic effect, okay? Example, the absorbance here is 0.1. After heating the DNA sample, it can be increased up to 0.8, 0.6, or 0.7, okay? So there's an increase in absorbance reading. And as shown in this figure, okay? So this is a typical melting curve profile of DNA depicting the hyperchromic effect observed on heating, okay? So the transition temperature TM here, okay? And from this point, expect that the DNA sample is double-stranded. After hitting the sample and you reach the TM, there's a gradual increase in the absorbance reading. You further increase the temperature and the absorbance increases. So at this point, the top one, expect that the DNA sample is completely separated or single-stranded. So this increasing absorbance, while increasing the temperature and separating the double-stranded into single-stranded, you called it as hyperchromic effect, okay? So that is the concept on hyperchromic effect. So what is the significance of knowing that nucleotides can absorb light, especially ultraviolet light? What is the significance of knowing that? That nucleotides, the bases, absorb light. Take note, the conjugated double bonds of purine and pyrimidine derivatives absorb light. The mutagenic effect of UV light is due to its absorption by nucleotides in DNA, resulting in chemical modification. Okay, you produce UV light-induced thymine, or pyrimidine dimer. And this may result to what condition? Too much exposure to UV light. What will happen?
Skin cancer.
Yeah, it will lead to squamous cell carcinoma or skin cancer. And remember, the absorbed UV light stays in our skin. Okay? Yun. Kaya mabilis tumanda, no? Maging old if you have too much exposure to UV light. And it may also result to skin cancer. And at pH 7, all nucleotides absorb light at wavelength 260 nanometer. Okay? It's almost the same range of the UV light. So remember, the 260 nanometer. And then another concept, you have the frameshift mutagenesis by DNA slippage. Okay? So during replication, okay? So this is actually replication. Because of the slippage, either you delete, okay? So this is 1, 2, 3, 4. 1, 2, 3, 4, 5. So this is you add another thymine here, okay? Or you delete here 1, 2, 3, 4, okay? So ilan na lang, sorry, this one pala, 1, 2, 3, 4. 1, 2, 3, 4 should be, okay? There's deletion of 1T, okay? So instead of 2, 4, 5, 6, you have only 4 here. So either deletion, rather addition or deletion, this may result to mutation or frameshift mutagenesis, okay? Because this affects the reading of your nucleotides if you either insert or you delete a particular nucleotide. Okay, so let's discuss now the chemical nature of RNA. Okay, so for RNA, okay, so again, the sugar in RNA is ribose rather than deoxyribose of DNA. And instead of thymine, in RNA, you have uracil. And most of the RNA molecule exists as a single strand. If there is a cap here at the 5' position and a poly-A tail, this is mRNA molecule. So that's how you recognize the messenger RNA. It has a cap and a tail, okay? Why is there a need for a cap and tail? Because the mRNA is produced in your nucleus, okay, from DNA, and it will travel from nucleus to the cytoplasm. So for its stability, it must have a cap. For its transport, it must have a tail, okay? And these are the different roles of different kinds of RNA. So tRNA or transfer RNA, this is the smallest among the RNA. Take note of this. It was asked in the board exam, okay? The smallest RNA, the tRNA, the function is transport amino acid to site of protein synthesis, okay? While ribosomal RNA, okay, it has several kinds. It combines with proteins to form ribosome, which is the site of protein synthesis, okay? And messenger RNA or mRNA, it is variable because it depends on the composition of DNA. it directs amino acid sequence. So if I ask you what is the template for protein translation, then your answer is mRNA. Okay. Okay, so this is actually the figure that explain the central dogma. Okay. So wala lang dito yung DNA to DNA. Okay. So this is transcription. You synthesize DNA into RNA. So the template for transcription is DNA. So you can produce ribosomal RNA from DNA, messenger RNA also from DNA, and the transfer RNA from DNA. So basically, this is transcription. Then once you have these transcription products, in protein synthesis or translation, you will be needing the proteins combined with ribosomal RNA forming your ribosome, which is the site for protein synthesis. The tRNA, which is the transporter of amino acid at the site of protein synthesis. And the mRNA, which will act as template for protein synthesis. So you have here the genetic information carried, okay, coming from your DNA molecule. Then this will assemble the mRNA, your ribosome, together with ribosomal RNA, and your tRNA carrying amino acid. And through this process, you will be producing proteins, okay? What happens to the tRNA and the ribosomal RNA? It will be recycled back. What happens to the mRNA? It will be degraded. So if I ask you what is the type of RNA that is usually hydrolyzed, okay? It is the mRNA, okay? It has the most rapid turnover. So again, mRNA is most frequently synthesized with the most rapid turnover among the RNAs, synthesized during transcription, and it carries genetic information from DNA as codon. So how many nucleotides do we have in a codon? You have triplet nucleotide. So meaning you have 3 nucleotides in a codon. And it is used as template for protein synthesis. Okay. It is again single-stranded and read and translated from 5' to 3'. To recognize mRNA, it has methyl guanosine cap at the 5' end and a polyadenine tail at the 3' end. So characteristic of tRNA, it is the smallest with the lowest molecular weight, also synthesized during transcription. And we have 60 different types. So 1 tRNA for every amino acid. Some amino acids have 2 or more corresponding tRNAs. It transfers amino acid from cytoplasm to the ribosome. It is also known as adaptor molecule. Okay. So it carries specific amino acid to the site of protein synthesis. Okay. This time, the tRNA contains the anticodon. So codon found in the messenger RNA, while anticodon found in the tRNA. It is single-stranded, but it can assume a secondary structure. So this is the only RNA that has the secondary structure. And the secondary structure of the RNA is known as the clover leaf structure. Another special characteristic of the RNA, it contains unusual or rare bases like ribothymine, pseudouracil, and dihydroxyuracil. The amino acid is always attached to the 3' end of the tRNA. And the 3' end of tRNA always end in CCA sequence. So that is very, very peculiar of the tRNA. The 3' end has the CCA sequence, and this is the point of attachment for amino acids. That's why we call the 3' end as the acceptor arm for the amino acid. And the amino acid is attached to the tRNA during the activation stage of translation. So again, this is the secondary structure of tRNA, which is clover leaf appearance. It has the unusual bases. It has the anticodon arm. So you have the anticodon here, which will interact with the codon in your messenger RNA. And then you have the acceptor arm that always ends with CCA, and this is the point of attachment of amino acid. Actually, the tRNA is also considered as a bilingual molecule. Why? Because it has nucleotide sequence, and in addition, you have the amino acid sequence. So meaning you have two languages carried in this structure. Kaya you called it a bilingual structure composed of amino acid and nucleotides. How about ribosomal RNA? Again, it is intimately associated with ribosome, the site of protein synthesis. It is the most abundant of all RNAs, but least characterized. So this is your ribosome. The total ribosome, 70S, S for Svedberg. We have the small and the large subunit. The small subunit is 30S and the large subunit is 50S. So when you combine the two, you have the total subunit 70S. And this is the ribosome in prokaryotic organism. So just remember, if it is prokaryotic, the numbering you need to remember for ribosome will be odd number, 30, 50, 70S. For the 30S subunits, what are the ribosomal RNA? You have your 16S ribosomal RNA. For the large subunit 50S, you have the 23S and the 5S ribosomal RNA. Okay. This one is the schematic presentation of a eukaryotic gene composed of exon, introns, exon, introns. So intervening will be you have the introns. And the coding unit in the DNA molecule is actually exon. This is what you need during transcription and translation. While introns are the non-coding units. These are the intervening unit. So when you transcribe DNA to RNA, you need to process the RNA that it must only contain exon. So introns will be removed via splicing reaction, okay? It's not slicing, SPL splicing reaction. So mature mRNA contains only pure exon, no more introns. Okay, so again, this is the DNA strands, the coding, and the template strand. So during transcription, when it is transcribed to DNA, it has to be anti-parallel. So if the RNA transcript runs from 5' to 3', then the template strand will be the bottom one, this 3' to 5'. So when you transcribe this, okay, so starting here, T, A, instead of T, it should be U because it's already RNA. AU, CG, AU, CG, TA. So this is what is being added during transcription. So this is the template strand. The top strand is also known as the coding strands or the sense strand. Why is it called as coding or sense strand? Because it has the same sequence with the RNA transcript, except for uracil. Instead of uracil, you have your thymine. So example here, A, same. U, this is thymine. G, U, T, G, A, G, CG, GCG. So it has the same sequence with RNA transcript except for uracil. It becomes thymine. So you can call it also as the coding strand or the sense strand. Well, this one, the template, another name is antisense strand. And then these are some of the species of the small stable RNA found in mammalian cells. Okay. Okay, so this is the DNA transcribed into RNA and then translated into protein. So again, for protein synthesis, you will be needing the mRNA as the carrier of genetic information. You will be needing the ribosome with your ribosomal RNA. And of course, not shown in the figure is the tRNA that carries amino acid. Take note of the polarity. The template here for transcription is the 3' to 5'. Then the RNA runs from 5' to 3'. The 5' correspond to the N terminal. Okay, this is very important. Kasi magkakabaliktad dyan if you will not take note of the polarity. So 5' to 3' correspond to the N to C terminal of the polypeptide chain. If prokaryotic ribosome has the odd numbers, the 30, 50, and 70, for mammalian or eukaryotic ribosome, you remember the even number, okay? So small subunit is 40, the big subunit is 60, and the total is 80S, okay?